Welcome to a fascinating journey into the world of cellular communication between bone and muscle. Recent advances in spatial transcriptomics have allowed scientists to create detailed maps showing how these tissues interact at the molecular level. Traditionally, bone and muscle were seen as separate entities, each with its own function. However, new research reveals that they work together as an integrated system, supporting movement, posture, and overall health. Today, we explore how scientists at Tulane University have used cutting-edge technology to uncover the intricate ways bone and muscle cells communicate, laying the groundwork for future discoveries in musculoskeletal health. Spatial transcriptomics is an emerging technology that maps gene activity directly within intact tissues. Unlike traditional genomic methods, which often lose spatial context, this approach preserves the location of gene expression, allowing researchers to see how neighboring cells interact. By applying spatial transcriptomics to a mouse femur and its adjacent skeletal muscle, scientists reconstructed cellular neighborhoods and communication networks. This breakthrough enables a deeper understanding of tissue maintenance, remodeling, and vascular support, and opens new possibilities for studying complex biological systems. Led by Professor Hong-Wen Deng at Tulane University, the research team generated data from 2,660 spatial spots across bone and muscle tissue in a young mouse. Their comprehensive analysis identified multiple major cell populations involved in tissue communication. Published in Bone Research, the study stands out for its depth and precision, offering a valuable reference for scientists interested in musculoskeletal disorders and aging. The combination of spatial transcriptomics and computational tools allowed for the reconstruction of cellular neighborhoods and communication networks, providing insights that were previously unattainable. Bone and skeletal muscle are often viewed as distinct tissues, but they actually form a highly integrated system. Together, they support movement, maintain posture, regulate metabolism, and preserve health. Scientists have long known that bone and muscle communicate through biochemical signals, but pinpointing where these conversations happen and which cells are involved has been a challenge. The new spatial transcriptomic map reveals that these tissues are linked through complex communication networks, reshaping our understanding of musculoskeletal biology. Study identified a diverse array of cell types participating in bone-muscle communication. These include osteoblasts, skeletal muscle cells, endothelial cells, immune cells, and stem cell populations. Each cell type plays a unique role in maintaining tissue structure, responding to physiological demands, and facilitating communication. By mapping these cells and their interactions, researchers have uncovered a surprisingly complex system that coordinates tissue maintenance and remodeling. Routine major signaling pathways were identified as key players in bone-muscle communication. Many of these pathways involve extracellular matrix proteins and growth factors, which help cells exchange information, regulate structural integrity, and respond to physiological changes. The organization of these pathways into distinct spatial networks suggests that tissue communication is not random, but carefully orchestrated by local cellular environment. One of the most significant discoveries was the identification of specific ligand-receptor pairs acting as molecular messengers between neighboring cells. These pairs include collagen-associated signaling between osteoblasts and muscle cells, thrombospondin-mediated communication involving immune cells, and VEGF-driven signaling supporting vascular function. Laboratory imaging confirmed the colocalization of several predicted molecular partners, strengthening confidence in the computational predictions. To ensure the accuracy of their findings, researchers validated key interactions experimentally and across independent datasets. Laboratory imaging confirmed the presence of predicted molecular partners within the tissue. Additional validation using mouse and human datasets supported many of the identified pathways, suggesting that some communication mechanisms are conserved across species. This robust validation process adds credibility to the study's conclusion. Professor Deng emphasized the importance of preserving spatial information in cellular studies. By maintaining the spatial context, researchers were able to uncover communication networks that would be difficult to detect using conventional sequencing approaches. This approach marks a significant step forward in understanding how cells interact within their native tissue environments, offering new perspectives on musculoskeletal health. Studies' findings have important implications for understanding musculoskeletal disorders such as osteoporosis, sarcopenia, and metabolic disease. These conditions often involve simultaneous deterioration of bone and muscle. By clarifying how tissues communicate, researchers can identify shared therapeutic targets and develop interventions that restore healthy communication. The reference map generated by this study will be invaluable for investigating changes in signaling networks during injury, aging, or disease progression. Understanding cellular communication pathways provides a framework for studying what goes wrong in musculoskeletal disorders. This knowledge may guide the development of targeted interventions that restore healthy communication between tissues. The study opens doors for collaboration across fields such as bone biology, muscle physiology, regenerative medicine, aging research, bioinformatics, and precision medicine. Spatially resolved transcriptomic maps set the stage for more precise diagnostic tools. By revealing how cells coordinate their activities through organized signaling networks, researchers can develop improved regenerative therapies and personalized treatment strategies. These advances aim to preserve mobility and quality of life, especially in aging populations, and offer hope for those affected by musculoskeletal disorders. Research delivers one of the first transcriptome-wide maps of bone-muscle communication, establishing a foundation for future investigations into musculoskeletal health and disease. Over time, these insights could contribute to more effective therapies and improved quality of life. The study's approach and findings are likely to inspire further research and innovation across multiple scientific disciplines. For those interested in learning more, several related studies have explored topics such as advanced osteosarcoma immunotherapy, muscle side effects from statins, synthetic grafting materials for bone cancer, and key proteins promoting bone formation. These readings provide additional context and highlight ongoing efforts to advance musculoskeletal research and improve patient outcomes. Summary: the creation of a spatially resolved transcriptomic map marks a milestone in understanding bone-muscle communication. By revealing the organized networks that coordinate tissue activities, this research paves the way for new diagnostic tools, therapies, and collaborative studies. As scientists continue to explore these cellular interactions, we can look forward to advances that will improve musculoskeletal health and enhance quality of life for many. The journey of discovery is just beginning, and the possibilities are vast.